Pandiyan. I'm from Biological Sciences. I'm the course director for this HEWB 128 course. It came to my attention. I don't want to start with a negative connotation, but I just have to talk about this. I heard about a little bit of attendance issue in this class. If you have any complaints or compliments about this course, it could be me. You can email me to tell about anything. But I also heard Dr. Weinberg talk about this, and on that day you had a test, and just right after this class, and some of you could not attend it. So the attendance is expected, although not required, but still it would make a lot of sense if you just follow the slides and review the slides until the regular attendance. Okay, so it's not like a big deal, but I just have to let you know that attendance is expected as well. All right. So if you have anything at all, questions about these slides or questions before your exams, please feel free to let me know by email. You can also make an appointment with me to talk to me. Come to my office. Anyway, I do research in immunology, so I have to deal with T-cell development, hypersensitivity, and autoimmunity. And my specific research projects involve HIV immunoregulation. So they have dumped a lot of topics. So I'm just going to give you key details about these concepts. So if you have more questions, as I told you, please let me know. And I'm also going to give you some homework for you to go and look up on these topics. But again, if you have questions, please discuss with me. All right? Okay, so this is the concept map. We're going to start with discussing about some of the oral lesions, inflammation in the oral mucosa, and what are the hypersensitivity issues or autoimmune manifestations in the oral cavity. And we're going to discuss about the causes, both the developmental defects and functional defects in the context of T cells that might be causing these oral lesions and inflammation. And then we're going to see the features of how a normal mucosa, oral mucosa looks like, especially touching upon immune balance concept and normal T cell development and functions and how these contributes to oral health. Okay, so usually the flow is from health to disease. I thought, let's start with disease and then progress to health. Okay, so that's the synopsis of the lecture today. So why immunology? Why do you think you need to be attending this class or body as post? Yeah.
Assuming the immune response is a large part of periodontal disease and we don't have any higher.
Yeah, and a lot of oral manifestations are also associated with systemic conditions, and as future dentists, you would be really better off. You can identify these oral manifestations and talk with the patient's physicians and bring about better point of care. Okay, so that's the whole idea of why we need to study immunology. And any oral immune disease that you know of, like I already put the slides there. So, like he mentioned, periodontitis. Any other?
Caries?
Caries, yeah. Well, it's okay. Okay, so one of the most common ones is gingivitis. It's it actually predisposes people to periodontitis. It is an unresolving inflammation of the gums, and it starts with plaque, and it's biofilm reaction or the unnecessary inflammation to this plaque that's causing this gingivitis, and it's T-cell mediated. Okay, so a lot of inflammatory cells can be found in the gingiva, and that's source of gingivitis. So it is a predecessor for periodontitis. Again, periodontitis is caused by bacteria or groups of bacteria found within this dental plaque biofilm. Okay, so gingivitis starts as an unresolving inflammation, but when it becomes more severe, where this inflammation is recruiting a lot more inflammatory cells, which are producing a lot more inflammatory factors that can even lead to bone loss, and that's where you have periodontitis. So it's a progressive destruction of periodontal ligament and alveolar bone that may result in tooth loss. Again, the initial perpetrators are the inflammation is again triggered by Th1 and Th17 cells. We're going to be looking at what these cells are, what Th1s are, what Th17s are. Maybe Dr. Levine talked to you about it, but we're going to talk about it in detail. So these T cells are, initially coming to the gingiva and starting this inflammation, and then they're bringing more macrophages and neutrophils, which can cause a lot of damage to the tissue, and that's where you have periodontitis. Again, periodontitis will be dealt by Dr. Weinberg in about 2 weeks in detail, but this is just an introduction about periodontitis. So one of the other manifestation that's caused by inflammation is oral lichen planus, and it's a chronic inflammatory condition that affects mucous membranes in mouth. Okay, so oral mucosa has layers of mucosal membranes covered by mucus, but when you have an appropriate or inappropriate reaction to the mucous membrane, then you have oral lichen planus, and it's characterized by swollen tissues, open sores, which can cause burning, pain, and other discomfort too. Okay, so again, the immune system here mounts an attack against cells of the oral mucous membranes. Nobody knows why this happens, but it's again, you can see a lot of infiltrating immune cells, T cells that shouldn't be there, but it's causing an inflammation. But right now, there's a lot of studies going on in terms of how local bacteria, actually triggering this inflammation, bringing these T cells and causing these inappropriate reactions. Okay, so the oral lichen planus patients, they also have a risk of developing oral cancer in the affected areas. So this is more severe form of inflammation called pemphigus vulgaris, and it's caused by lack of adherence of epithelial cells. So epithelial cells, did someone talk about the oral epithelium before? Yeah, so oral epithelium, there are multiple layers of epithelial membranes, and these epithelial cells are attached to one another but through structures called desmosomes. Okay, so these desmosomes are, they maintain the structure of epithelium intact. But in this case, there are some autoantibodies. Antibodies are produced by B cells. Here IgG, IgM, and IgA antibodies are produced against these desmosomal structures, and that leads to loss of this epithelial barrier, or the epithelial cells get loosened, and because of that, there is an infiltration of a lot of bacteria, commensal bacteria, into the mouth, and that causes the inflammation. Okay, so normally it looks like you can treat it by corticosteroids and other immunosuppressive drugs, but still, not much of research is going on, what is causing this. Okay, so, and it looks like a genetic defect of what, these antibodies, or maybe a prior infection might have caused some antibodies that are cross-reacting to the desmosomes. So this is again, it's more of an allergic reaction to antibiotics, or sometimes herpes simplex virus infection can cause this. It's called erythema multiforme, and it's acute onset of blisters and ulcers of skin and oral mucosa. So it's a hypersensitive reaction, or an allergic reaction to antibiotics, or probably antibiotics can cause some imbalance of microbiota in the oral cavity, and that leads to the progression of inflammation, which can be severe and acute because it's a hypersensitivity reaction. So SLE, or systemic lupus erythematosus, is another disease. It's not primarily in oral mucosa. It's a systemic disease, but you have a lot of oral manifestation like mouth ulcers and sores, and this inflammatory sores can, a lot of times can progress into oral cancer. Okay, so that's why this is serious. And so SLE is caused by, again, autoantibodies to some antigens in kidney, and it's characterized by fever, weight loss, and butterfly rash across the nose and cheeks. Okay, so again, it's mediated by antibodies produced by B cells, but T cells also help in this process. So Sjogren's syndrome, you might have heard about it. It's a very common autoimmune disease. About 3 million Americans have this disease, and about 90% of the patients are women. Sometimes it's associated with rheumatoid arthritis. It's again caused by CD4 T cells. T helper cells are CD4 T cells. Sometimes I call them T helper cells, but they are the same as CD4 T cells. Okay? And also B cells. So here, Sjogren's syndrome, people have antibodies and T cells attacking salivary glands, and this leads to the dryness of mucosa. Okay, so as you all know, salivation is very important mechanism, innate mechanism of keeping the infections in check, because salivary flow keeps out a lot of pathogenic bacteria and fungus. So because these people have lack of salivary flow, they have experience in eating, and their tongue is actually having a cobblestone appearance, and they also have taste and speech disturbances. So, and because of lack of saliva in the tongue, they have increased dental cavities, streptococcal infections, and candidiasis, fungal infections in your mouth. And Crohn's disease, it's one of the inflammatory bowel diseases that also has oral manifestation associated with it. So it affects your oral cavity in about 8 to 29% of the patients who have Crohn's disease, and it is characterized by swelling of gums, ulcers in mouth, and swelling of lips, and they also have eating difficulty. So it looks like these oral sores appear much before the Crohn's disease or inflammatory bowel diseases. So, it might be a great way to diagnose that these people are having initial symptoms of inflammatory bowel disease. And, so some of the recent research has shown that a lot of the people that are having inflammatory bowel disease, they also have periodontitis. Okay, so so they have a connection basically, and looks like the perpetrators are T cells, because in Crohn's disease, T cells are the ones that are affecting the gut, and they're causing inflammation in the gut, and it's the same in the oral source as well. So oral candidiasis, it starts as a fungal infection. It's not an inflammation per se, but once it's recurring and when it leads to a chronic inflammatory state, then it's an inflammation. Otherwise, it's an infection. So initially, as an infection, it's called thrush or pseudomembranous, where the fungal growth is very superficial, and and it appears as white plaques. However, erythematosus and angular cheilitis and chronic hyperplastic candidiasis are the manifestations where you have chronic candida infections in your mouth, and that's associated with loss of because of immunodeficiency, like HIV positive patients, because of their lack of CD4 T cells, they have this recurrent candida infection when they are not treated properly. And also people undergoing steroid treatments or cancer treatments, because of the chemotherapy and immunosuppressants, they have these oral candidiasis. So normally, candida is present in like 60% of the healthy individual as a commensal, but in the context of immune deficiency or immunosuppression, these can cause chronic inflammation in the mouth. So again, it's caused by loss of T cell functions, for example, during an HIV infection, and loss of immune regulation, for example, during cancer treatments, where a lot of regulatory T cells are lost. Okay, so as you all know, immune system in the oral cavity is part of the mucosal immune system. So we're looking at this oral inflammation in the context of oral mucosal responses, because oral mucosa is part of the mucosa, and then we should be looking at the mechanisms in the context of oral mucosa. And it shares several features with other immune systems. Do you know what the types of immune systems are? The major types of immune system? Oh, innate, adaptive. Okay, so even more general than that. Okay, so it's just mucosal immune system, cutaneous immune system, systemic immune system. So mucosal immune system are like, gut, vaginal, and oral mucosal immune system. Cutaneous is skin immune system, where in a skin has a lot of resident immune cells, like distinct immune cells, different from mucosa and systemic immune system. And systemic immune system is like basically blood immune system. Okay, so these are distinct because of several features that we'll discuss about. So mucosal immune system is consists of respiratory, gastrointestinal, and glandular immune system. Okay, so here conjunctiva is eye-related mucosal immune system, and then this is respiratory. Sinus, trachea, and lungs, they have respiratory immune system. And it's so unfair that they've included oral cavity with gastrointestinal immune system, but it should be classified as oral mucosal immune system. Maybe in a few years we'll change that, we'll see. But gastrointestinal mucosal immune system consists of esophagus, stomach, and intestine. And glandular immune system are formed by various glands all over the body, including urinogenital immune system. So oral mucosa, like why I said it should not be categorized under gastrointestinal, is because its epithelium is different. It has a distinct structure compared to gut. Okay, so for example, did you go through gastrointestinal mucosal immune system already? Did Dr. Levine touch upon that? No? Okay. So anyway, usually mucosal immune system consists of primary organ, the tissue organ, here in this case gingiva, tongue, and the palatal epithelial immune system. That's the immune system associated with the oral tissues themselves, and also associated lymphoid tissues. Okay, so they are like for oral mucosal immune system, it is adenoid, palatine, and lingual tonsils. Okay, so these tonsils are lymphoid tissues for oral mucosa, and these are called mucosal associated lymphoid tissue, or malls. Okay, so every mucosa is associated with structures called malls, or these mucosal associated lymphoid tissue, and for oral mucosa, it's these different tonsils that are associated with oral mucosal immune system. Okay, so and this is distinct from what you find in respiratory tract and gastrointestinal tract. And so it's important to study mucosal immunity because mucosal surface is the first line of defense. Okay, so mostly like 90% of the infections start with the mucosal immune responses because it's the first line of defense. So anything that you come across, any new antigen that you come across, has to go through the mucosal immunity first, and then it just is seen and taken care by the systemic immune system. So, question, because whatever you ate, like food antigens, I mean food has a lot of antigens, a lot of protein, carbohydrates that can be potential antigens. Do you think your gut is making an adaptive or any immune response against what you ate for lunch? Because it's a mucosal immune response, and we have mucosal immune response, and we have a lot of antigens going through the mouth and the gut. Do you think our immune system is acting against these food antigens? No, why?
You've probably eaten them before, so they're not unfamiliar.
Okay, so that's a process called mucosal tolerance, okay? So you don't have a strong response, but there's something going on, okay? But the immune system says, okay, this is an innocuous or harmless antigen, this is a food antigen that we have seen multiple times, so we're not supposed to generate an immune response, okay? So that process is called oral tolerance. That happens in the mouth, that happens in the small intestine, that happens in the colon, where your immune system is being tolerant to what it sees as innocuous or harmless antigens. It also includes commensal microbiota that we find in the oral mucosa and the gut, okay? So that process is called oral tolerance, and that's very, very distinct from systemic immune system. So systemic immune system, whatever it sees, it will mount a response, and that's what it's designed to do. But here in oral mucosa, it is also designed to be tolerant to certain antigens if it's harmless, okay? So that's where the distinct features of systemic versus mucosal immune system are coming in. Okay, so systemic immune system normally functions in a sterile milieu, but mucosal immune system functions in unsterile environments, because a lot of the food that you can eat can be unsterile, okay? So, but still, we don't mount a bad immune response. And systemic immune system can afford to respond to invaders with highest intensity, whereas mucosal immune system respond to broad range of antigenic stimuli, which means, sometimes it'll respond in a really intense way, sometimes it will respond in a very mild way, depending on the pathogenicity or the ability of the antigen to cause the disease. Okay, so we look at how these are taken care of mechanistically in a while, but these are the differences. So MOLD, as I said, they have specific mechanisms to do this, like to generate a wide range of antigenic stimuli or antigenic responses. Okay, so this is happening because we have to avoid bystander tissue damage and immunological exhaustion, because you don't want our immune cells to just keep responding or fighting the food antigen or commensal, because that's going to lead to exhaustion. So we have immune responses or regulatory immune responses to prevent excessive or unnecessary immune responses from happening. So that's the part of immune regulation. So, so these are again distinct features between protective immunity and oral tolerance that we just talked about. Here, for protective immunity, the antigens are invasive bacteria, viruses, toxins. Oral tolerance, the antigens are food proteins and commensal bacteria, for example. Here, protective immunity, for example, in the gut and the oral mucosa, you have intestinal IgA. Specific antibodies are present in the serum all the time, but in the case of tolerance, you have only local IgA, which are actually not present in the serum, but they tell the immune system that you're not supposed to respond to this, okay? So that's because these are immunomodulatory IgAs and not immunostimulatory IgAs, okay? And they're not present in the serum. And the T cell response is local and systemic effector and memory T cells, like normal. But here in oral tolerance, you don't have an effector T cell response, because this leads to immunomodulation and not immunostimulation. And for protective immunity, when these are re-exposed, they have an enhanced immune response, which is the basis for vaccination. But in oral tolerance, it's low or no response. So once they know this is a harmless antigen, they won't respond to it anymore, okay? Because immunomodulation has already been achieved. Okay, so some of the mediators you might have seen for these that distinguishes immune tolerance versus immune stimulation or E cells, neutrophils and macrophages, components of innate immunity, and natural killer cells, and T cells for adaptive immunity, and dendritic cells. They act as a bridge between innate and adaptive immunity. Okay, so when you don't have this tolerance to innocuous antigens or self-antigens like our own protein, that's where we have immune balance. Okay, so when there is a balance, under healthy situation, your immune regulation and host immunity are, in a perfect balance. That's why it's balanced. But when you have too much of regulation, that leads to imbalance, and when you have too much of immunity, again, that leads to imbalance. Okay, so when you have too much immunity, it leads to inflammation, and when you have too much regulation, you have impaired host immunity. Okay, so and this imbalance is caused by a variety of factors. For some people, it's genetics. They have mutations that leads to this imbalance, which is why some people are, some polymorphisms are associated with increased autoimmunity in some people or some population. It can also be environmental or chronic infections that people have been exposed to, or microbiome dysbiosis. For example, if you are in constant antibiotics for some reason, your microbiome gets dysregulated. That can also lead to this imbalance in your immune system. So here the threat can be both external or internal. When there is an immune overreaction, that leads to autoimmune problem. If the threat is internal, that means your antigen is coming from inside. Okay, so it's one of your tissue-specific antigens or your organ-specific antigens. That's an internal threat that leads to autoimmune problem. For example, type 1 diabetes, arthritis, and IBD are caused by immune overreaction when there is an internal threat. An allergic immune reaction is caused by an external threat. For example, a pollen allergy or peanut allergy. They're caused by external threat due to immune imbalance. Okay? An immune underreaction is caused by, leads to cancer or infection, depending on whether the internal threat is a cancer, that leads to cancer, or the external threat is probably an infection, and an immune underreaction is not a good thing to happen at that point. Okay, so we need a balanced immune system with an optimal effectiveness for this immune balance to be in proper place. But when you don't have it, which is why, which is when we have hypersensitivity and autoimmune reaction. Okay, so there are types of autoimmune reactions: type 1, type 2, type 3, type 4, DTH or delayed type hypersensitivity, or chronic inflammation and organ-specific autoimmunity. Okay, so here these are all different types of reaction which shouldn't happen normally, but it still happens because of the immune imbalance that we were just talking about. Okay, so this is an example of an allergic test, type 1 hypersensitivity immune reaction, where locally antigen is administered, very, very small amounts of antigen, and you can see this local immune reaction. It's a skin test, but that's an example of type 1 hypersensitivity reaction. Okay, so you can go through this slide later on, because I don't believe in dumping the slides and letting you read all that. You can read later on. Let's just go to more of a discussion. But if you have questions in that slide, please let me know. Okay, so this is with a picture. I like pictures than words. So let's go with a picture. This is a type 1 hypersensitivity reaction, and it's mediated by IgE, one of the antibodies, and it's where antigen or an allergen induces cross-linking of IgE that can bind to mast cells or basophils. And so this cross-linking, along with, for example, this is an allergen, for example, a pollen, this cross-links these antibodies and it binds to the Fc epsilon receptor on the mast cells or basophils, and these are capable of releasing a lot of vasoactive peptides, which also includes histamines. Okay, so that leads to release of histamines leads to allergic reactions. Okay, so it's basically starting with the IgE binding to the Fc epsilon receptor of these allergy-causing cells. Okay, that leads to degranulation. Okay, and examples are some allergic reactions: hay fever, eczema, and hives. These are all the examples of type 1 hypersensitivity reaction. So this is type 2, and that's caused by IgG and IgM. So there are two types of reactions that are that result from IgG or IgM binding to the antigen. In this case, it can be an antigen that is recognized by the body as foreign. Okay, so and that binds to the antibody, and one hand this antibody binds to the Fc receptor of a cytotoxic cell. Cytotoxic cell is able to produce cytotoxic granules like granzymes and perforins, and once you have this is called ADCC, where an antibody and antibody receptor are brought to the site of cytotoxicity, and this release of perforins can kill the cell. Okay, so here it's mediated by IgG that's bringing the ADCC in place, and the other way is the antibody is binding to the antigen and it's activating the complements. Okay, so the complements can also lead to lysis of the cell that's expressing the antigen. Okay, so here you have two things happening. One is ADCC, which is antibody-dependent cytotoxicity, and the other one is complement activation, and both of them are initiated by IgG and IgM antibodies binding to the antigen. Okay, so here the blood transfusion reactions is an example of type 2 hypersensitivity reaction. Okay, so this is type 3, where you don't have antigen binding to any cell or being expressed by any cell. The antigen is in the soluble form in the blood, but it forms complexes with antibodies specific to this antigen, and this is again, these immune complexes are recognized by the complements, and that leads to the activation of neutrophils and other type 3 cells. Okay, so this is caused by immune cells, that's type 3, and so these are the manifestations that occur due to type 3 hypersensitivity reaction. So far all of these are antibody or innate cell mediated, but type 4 is T cell mediated or adaptive immunity mediated. Okay, so here the immune mediator is T cell, because the T cells get sensitized to the antigen directly. Okay, so mostly the T cells that are causing allergy are these type of hypersensitivity reaction are Th2 cells, but Th1 cells can also can cause hypersensitivity type 4 reaction. Okay, so here the sensitized T cells, once they are activated, they can secrete cytokines that can activate the macrophages, and also neutrophils that can cause tissue damage directly. Okay, so these are DTH or hypersensitivity 4 reaction. Okay, to go into detail, please look upon on early and late responses of type 1 hypersensitivity symptoms and genetics of asthma and allergy. Okay, so this is in QB immunology. You all have access to QB immunology, right? Yeah?
Is it through the prejudice source?
Yeah,
Oh, okay.
yeah, yeah. So let's go into organ-specific autoimmune diseases now. So there are several autoimmune diseases. These are a list of organ-specific autoimmune diseases, and these are non-organ-specific autoimmune diseases that we, some of them we just went through just now. Again, Approximately 23 million people in the United States are afflicted with at least one autoimmune diseases, and many of them have some associated oral symptoms as well. And women are at great risk of acquiring an autoimmune diseases than men are. It's so unfair, but that's true. And heredity, gene mutations, microbes and hormones drive the onset and symptoms of these diseases. And most body parts can be affected by these diseases. And there are treatments, mostly corticosteroids and immunosuppressive drugs, but still there are no cures. Okay, so some key points to remember about autoimmunity are this happens when immune system goes haywire. The normal purpose is to defend the body, but sometimes they can attack itself. That's autoimmunity, and that's called, against self-antigens or self-tissues or organs. And even during normal immune responses, overreaction of the immune system can cause inflammation. Okay, so it's not like a inappropriate antigen they are responding to. They are responding to proper antigen, for example, an infection. But usually the immune system or immune response is controlled. But when you don't have a control, when there is an overreaction or exacerbation, that also leads to chronic inflammation. Okay, and most of them are T-cell mediated, and some are autoantibody or B-cell mediated as well. Okay, so just to give you an example, immune cells are like cops keeping the criminals or pathogenic microbes at bay, but sometimes cops can show aberrant behavior, and this is an example of that. Okay, so autoimmunity was first discovered by Paul Ehrlich in 1908, and he got a Nobel Prize for that, and he put forth, so he called autoimmunity as horror autotoxicus, where, it's similar to the scorpion stinging itself, okay, so, and he said a well-adapted contrivance, regulatory contrivance or immunoregulatory mechanism should be there to counter this problem, and we do have it, okay? So let's talk about that. So this is sort of the structure of immune system, innate, adaptive immune system, where you have a lot of innate cells, myeloid cells and lymphoid cells, and lymphoid cells are T and B cells. These are main perpetrators of autoimmunity, so we're going to be looking at them in detail. So these are two classes of lymphocytes, the main culprits of autoimmunity, and T cells are mainly concerned with cellular immune responses to pathogens such as bacteria and viruses. They also regulate the responses of B cells and overall immune responses, because a class of T cells, CD4 T cells, can help B cells to produce the antibodies. These are helper T cells. And the other class is cytotoxic T cells, which produce granzymes and perforins. They actually kill the infected cells, or they kill the cancer cells. Okay, so these are called cytotoxic T cells. And B cells are responsible for production of antibodies that act against existing pathogens normally, but in the context of autoimmunity, they produce all antibodies that are abnormal. Okay, so normally T cell activation starts with an antigen presentation, like you probably read about, MHC and antigen presentation in Dr. Levine's class, but so this is the overall antigen presentation and activation of T cell. And so once there is an antigen presentation, once a T cell recognizes an antigen through the T cell receptor, the first thing it makes is IL-2, okay, so, or interleukin-2, a cytokine that's responsible for the proliferation and activation of these T cells. Okay, so they get activated, they proliferate, and they take care of the bacteria or fungus and mediate this host protective immunity. Okay, so this is a very simple form of activation of T cells, and we are going to see how this becomes abnormal later on. Okay. So when this antigen is producing or is just inducing T cell activation normally, it becomes a Th1 cell, okay? So it's always the context of this activation that determines whether these T cells are going to become T helper 1 cells or T helper 2 or T helper 17 or whatever they are, okay? So the context or the cytokine milieu of this, like during the antigen presentation, influences how the T cells are going to differentiate into different types of helper cells, okay? So here are the main types of helper cells. Here, so in the first case, when a naive cell gets a T cell receptor stimulation through an antigen in the context of IL-4, it becomes a Th2 cell or T helper type 2 cell that can produce IL-4 and IL-5, and these cells are responsible for causing allergic and anti-helminthic responses, okay? So these are normally anti-parasitic cells, but then they can also cause allergic responses, okay? And Th1 cell is what I just said. This is the normal context where T cells are activated in the context of IL-12, interferon gamma, and IL-18, the normal pro-inflammatory cytokines. And under this context, they become Th1 cell, and they can make interferon gamma and TNF alpha, okay? So these cells take care of the intracellular bacteria by activating macrophages and, basically destroying these infected cells, okay? However, these cells, Th1 cells, are also important for inadvertently can cause chronic inflammation, for example, in periodontitis or inflammatory bowel disease, because you have excessive responses of Th1 cells there, okay? And when the T cells are activated in the context of these cytokines, this is a set of cytokines like IL-1, interleukin-6, IL-23, and TGF-beta. So when you have these in the context of your epithelial cells or produced by dendritic cells in this milieu, then these naive cells can become Th17 cells, which means that they can express cytokines such as IL-17, IL-22, and these normally take care of antifungal and extracellular bacteria, but they can also predispose your cell to inflammation. Okay, so 10 years ago, everybody thought it was just the Th1 cells that is causing a lot of inflammatory diseases. But right now, everybody is thinking, okay, it's a combination of these culprits, Th1 and Th17 cells. Okay, so that's causing the inflammation in your body. Okay, so whenever there is an excessive response of Th1 and Th17, you have inflammation. Okay, so or chronic inflammation. And apart from all these immunostimulatory cells, we also have Treg cells or regulatory T cells. Okay, so these do opposite to what these guys do. Okay, here a naive cell gets actually activated in the context of IL-2 and TGF-beta. When that's happening, these cells express a protein called FOXP3 that shuts down a lot of inflammation or inflammatory processes in T cell. So they can only produce certain immunomodulatory cytokines, so they can regulate each of these cells. Okay, so normally we have these Treg cells achieving immunomodulation. That's why most people are healthy. But when you have defects in these cells, that's where you have excessive inflammation caused by Th17 or Th1 or Th2 cells. Okay, so these are immune regulators and these are immune stimulators. So we just talked about these cells, Th1 cells, they destroy intracellular bacteria, but they can cause inflammation. Th2 cells, they they help making antibodies in B cells, and they neutralize helminths and parasitic microbes, but they can also cause allergy. In Th17, they have cell-mediated immunity and may cause may help defense against fungi, but they can also be pathogenic. And T follicular cells, these are present, they are like Th2 cells, but they are present in the germinal centers of spleen and tonsils. So regulatory cells, again, we are reviewing what we just talked about. The markers of these cells are CD25 and FOXP3, and they actually regulate or control immune responses to limit reaction against cell tissues, okay? So these are Tregs or regulatory T cells, okay? And these is one of the important subset of cells important for critical for immune tolerance. So immune tolerance, like we said, like we talked about, there are two types of tolerance: central tolerance and peripheral tolerance. Central tolerance is like choosing the right cop, and peripheral tolerance is like proper training during the job to prevent any reaction against normal people or any abnormal behavior towards, to prevent so that they can distinguish between criminals and normal people. Okay, so they can protect harmless people or tolerate harmless people. Okay, so central tolerance is like choosing is at the level of T cell development, and peripheral tolerance is during the activation and changing cytokines during the activation. Okay, so central tolerance, like I said, is happening during the development, choosing the right cop, and it happens in the thymus. And central tolerance is happening at two levels, one through positive selection and another through negative selection. Okay, so positive selection is selecting good cops, and negative selection is disqualifying potentially bad cops. Okay, so I'm just trying to give an example so you can remember, because you might have questions in the exams about this. So I am just trying to give you some, clues here or examples here. So thymus is where the T cell development is happening, and thymus is present right here between the lungs. So that's the cross section of the thymus. And this is an electron micrograph where the green T cells are actually present in the cortical epithelium. So these are actual T cells under the electron microscope, and that are developing and coming out of the thymus. So here, the thymus is consisting of two distinct regions: the medulla, inner region, and the outside cortical region. So this is where the T cell development happens. It has two distinct steps. One is early thymocyte development, where the T cell receptors are developing and rearranging, and the second step is the selection process, where you have positive selection or choosing of the right cops, and the negative selection, disqualification of the wrong cops, potentially wrong cops. Okay. And then it's followed by exit of mature T cells that are, potentially going to be acting against the pathogen, but not going to be acting against cell tissue or not going to cause any inflammation. So now you can look through the slide later on. So it basically talks about, where what happens. So first of all, the lymphoid progenitor, common lymphoid progenitor, comes through the blood into the corticomedullary junction, and then it enters through the subcapsular zone. It enters the cortex, which is where most parts of the T cell development happens, and then it goes to the medulla for the second process of selection, or selective processes happen. Okay, so let's just so here, this is a T cell that's in the cortex. So it went through all the double negative, double positives. You know what double negative, double positive cells are? Okay, so T cells are basically either, like we just talked about, CD4 or CD8. So these are single positive, or SP, CD4 single positive, or CD8 single positive cell. But when they come out of when they're in the first stages of development, they are double negative. They don't express either of these receptors, CD8 negative, CD4 negative, which is what is called double negative. Okay, so that's double negative. But then they become double positive. They express both the CD4 and CD8 co-receptor, and that's when they are called double positive stage. Okay, so that's basically based on the expression of their CD4 and CD8. Okay, whether they are double negative or double positive. So when they are double negative or double positive, they also express other molecules that are important for each step of these processes. But all you have to remember is at the double negative stage, when they are not expressing both CD4 and CD8 receptor, that's when their beta receptor rearrangement is happening. Okay, so we're talking here about alpha beta T cell receptor positive CD4 cells or CD8 cells. That's what we call as T cell development. Okay, so that's what is going on in the thymus. So when they develop, their T cell receptor, first beta receptor gets rearranged, and and once that beta rearrangement is stable and their alpha receptor is also rearranged, and then they have we have a T cell that is expressing a certain alpha receptor and a certain beta receptor. Okay, so that's when they're undergoing this selection processes. Okay, so most of these happen in the cortex, and they go to negative selection process as single positive CD8 cell or single positive CD4 cell. Okay, and then there is another process of selection that we will just see here in the next slide. Okay, so in the cortex you have in this case CD8 and CD4 expressing cells, and most of the cells, they have an alpha and beta receptor that's not recognizing anything. Okay, so that's not recognizing any antigen in the thymus. So that means they will die by neglect. Okay, so that happens during the positive selection, because they don't express a functional beta receptor or alpha T cell receptor. Okay, so that happens, that process is called death by neglect. Okay, but then there is a subset of the cells that are expressing very, very high affinity and T cell receptor that can respond to all these self antigens in a very high affinity manner. That means when these go to the periphery, they are going to respond to your own cell tissues. Okay, that's not good. Okay, so that's why these cells are also eliminated in the thymus. But the cells that are having intermediate affinity, that the immune system thinks that these are not going to cause any inflammation in the periphery, they get positively selected. Okay, so this is called positive selection, where very low affinity TCR carrying T cells and very high affinity TCR carrying T cells are excluded in the thymus, and only cells that are carrying intermediate T cell receptors are selected and goes to the medulla. Okay, so this is the process of positive and the negative selection. And again, in medulla we have a specific cortical epithelial cells that express a protein called AIR, and these can also express a diverse array of self-antigens. Okay, so again, these single positive CD4 T cell or single positive CD8 T cell, if they are carrying high affinity T cell receptors that can recognize these self-antigens that are expressed by AIR positive epithelial cells, they are again getting deleted. Okay, again, another subset of dendritic cells say, make sure that this is happening again. So all the T cells that are carrying the cell-specific T cell receptors are deleted in the thymus. Okay, and this process together is called negative selection, or where the COPs are disqualified because they can potentially be harmful to your own self-antigens outside. Okay, your own cells of the body or the tissues in your body. Okay, so that's why these cells are deleted in the thymus, and you don't have self-reactive T cells coming out of the thymus. So it's an antigen-dependent selection process. And the affinity to self-peptides actually drive the selection process. Like we said, if the affinity is too high, they just get deleted by negative selection, and if the affinity is too low, which is the case for most of the cells, they die by neglect. Only the cells that are having intermediate affinity, but still they don't actually react to your self-antigens under with a high intensity, only those get selected and come to the periphery, okay? So that also means that there is a potential ability for these cells to respond to a cell in a higher affinity later on, okay? So mostly it doesn't happen in healthy individuals, but in autoimmune people, because these are carrying intermediate affinity to your self-TCRs or self-antigens, they are causing autoimmunity. But 95% of the times it doesn't happen, but 5% of the times it does happen, okay? But in healthy people, the selection process makes sure that we don't have an immune response to your self-antigens. Okay, so this is where mutations come into play. For example, some people, we just talked about the medullary thymic epithelial cells, MTECs, and these express a transcription factor called AIR that can actually lead to transcription of or expression of a lot of self-antigens, and that is involved in this negative selection process. But some people have mutation in this protein called AIR, okay? So they don't get to express all the self-antigens, so the selection process or the central tolerance process is kind of defective in these people, okay? So AIR deficient people or people who carry mutations in this protein have a lot of autoimmunity going on, okay? So that's a defect in central tolerance, okay? So here it's caused by a mutation in a protein called AIR, and these patients have an autoimmune disease called IPSID or autoimmune polyendocrinopathy and ectodermal dystrophy, okay? Like I said, this is because of the mutation in AIR protein, and it's normally expressed in medullary epithelial cells of the thymus, and which is taking care of the central tolerance, but because of this mutation, they don't have the central tolerance process. So, and because of that, they have a lot of autoimmune manifestations in because of the responses against antigen present in endocrine organs, adrenal cortex, gonads, and pancreatic beta cells. Okay, so they have T cells that can respond to all these cell tissues, which and leads to pathogenicity or autoimmunity in these people. Okay. So now we'll go to peripheral tolerance, which is not related to T cell development. It's like I said, it's proper training during the job for a cop. So we're going back to reviewing our slide about the T cell activation, where an antigen presentation can lead to an activation of T cell, production of IL-2 and other cytokines. When it's not regulated, then you have an excessive immune response that can lead to chronic autoimmunity and inflammation. Okay? So this process should be regulated, and how it's regulated is by several intrinsic features or proteins expressed by these T cells, such as CTLA4, TGF-beta, IL-10. Okay? So these are immunomodulatory proteins that can block excessive immune responses by acting on antigen-presenting cells or blocking the production of inflammatory cytokines, okay? But a lot of times what happens is once you have a response to a pathogen, most of the cells will die of apoptosis, okay? So that's just basically death, okay? If you don't have this death happening at the right time after an infection, these pathogenic cells are the cells that can potentially produce a lot of inflammatory cytokines, can still go around in the body causing inflammation. So you have immunomodulatory cytokines and apoptosis of T cells that blocks these T cells from causing autoimmunity. Okay, so that's a way of regulation or intrinsic regulation in T cells that prevents autoimmunity from happening. So that's one of the peripheral tolerance mechanisms. And the other thing is extrinsic, where the regulatory T cells that also came from a naive cell differentiated in a different milieu. These cells can produce a lot of these immunomodulatory cytokines. They also expand along with these cells, but they, instead of producing pro-inflammatory cytokines, they produce anti-inflammatory cytokines, and they prevent this excessive expansion or excessive inflammatory cytokine production in these cells. Okay, so we have intrinsic mechanisms and extrinsic mechanism by regulatory T cells to prevent this from happening. Okay, so these are regulatory processes that are part of peripheral tolerance mechanisms. So this happens at different levels. Like this peripheral tolerance mechanism leads to balance of lymphocyte populations between the pro-inflammatory and the anti-inflammatory T cells, and they also regulate local antigen-presenting cells and phagocytes to prevent tissue damage, and they also produce enough tissue factors to achieve immunomodulations in the tissue, and they also produce immunomodulatory cytokines such as TGF beta and IL-10 that I just mentioned to achieve peripheral tolerance in tissues. Okay, again, Tregs are also present in tissues to be doing all of these. So what are Tregs doing? Tregs are, like I said, these are FOXP3 positive cells, and they have different mechanisms by which they control excessive inflammation or excessive expansion of inflammatory cells. One is production of anti-inflammatory cytokines such as these, and the other is IL-2 consumption. Okay, I said IL-2 is an activation factor or expansion or required for expansion of T cells, but when this is consumed by regulatory T cells, it's causing a competition and leads to apoptosis of potentially pathogenic T cells. Okay, so that's one thing. And the other thing is they can also directly kill the cells by producing cytolytic factors such as granzymes. So these are three important features of Tregs, or how they're regulating the T cells in the periphery. So to review that again, FOXP3 positive cells or Tregs can produce inhibitory cytokines. They can inhibit antigen-presenting cells. They can cause they can cytotoxically kill inflammatory cell. They can also deprive the inflammatory cell of the survival cytokine IL-2 because they are expressing the IL-2 receptor. Okay, so these are the four major things that they achieve the peripheral tolerance by regulating inflammation. Okay, but there are people who have mutations in FOXP3 genes, and these are IPEX patients. Immune dysregulation, polyendocrineopathy, enteropathy, X-linked syndrome, and these people have mutations in FOXP3, and it leads to immune destruction of bowel and pancreas, thyroid, and skin. They often die in the first 2 years of life due to sepsis and failure to thrive. And obviously they don't have Tregs, or they don't have functional Tregs. And this is a very important example of a defect in peripheral tolerance. So to review again, central tolerance mechanism prevents autoreactive cells from coming to the periphery, and peripheral tolerance mechanisms prevent overreaction of immune cells during normal immune responses to infections or cancer. So that achieves that, so good coughs remain effective and harmless to regular civilians. So what could be the other reasons for unintended inflammation? We were talking about it a little bit, other than just T cells and the immune system itself. We did talk about it a little bit. So resident microbes or good bugs. Okay, so normally the common cell microbiota that we have are beneficial in many ways. Okay, but they can become pathogenic if there is a dysbiosis in this bacteria, both in oral cavity or the gut, wherever. Okay, so these are normally harmless, but they're potential criminals. So we have more prokaryotic organisms or microbiota in our bodies than we have eukaryotic cells. For example, in the gut we have 10 times 14 microbes. And in fact, only 1 out of 10 cells in our bodies is human, other 9 come from these bugs. So they have a very important or very critical influence on how our immune system and how our metabolism works. Okay, so that's why when there is a dysbiosis, there is a change in the immune system or change in the immune cells, in the local immune cells that causes inflammation. So, resident microbial communities or human microbiome consists of various bacteria. They are present in skin, vaginal, and gastrointestinal tracts. And it was discovered in 2000s where a change in the microbiome was shown to be important for whether you're going to be obese or lean. Okay, so that's when research, it caught the attention of researchers and people have started working on the microbiome, and now it seems it affects almost everything, not only in the immune system, about every part of your life. Okay, so they're very important. And in oral mucosa, there's about 700 bacterial species, fungi and viruses. So, if there is a balance in each of these, for example, even periodontal inflammation is now considered to be an imbalance of the commensal microbiota. It's not caused by, those keystone pathogens. They're always there. It's just that the balance is affected and it's causing inflammation. Okay, so normally they are there for pathogenic, for preventing pathogenic invasion and also preventing inflammatory disorders. But when you cause dysbiosis, for example, changing your microbiome by repeated use of antibiotics, for example, it can lead to dysbiosis that can lead to inflammation. Okay, so normally they are important for mucosal tolerance, but at the same time, if you cause dysbiosis in these bacteria, it can also lead to inflammation. Okay, and the precise immune mechanisms are still an active research area, including some studies in our lab, but in the context of oral microbiota, a lot has to be studied. So, inflammatory bowel disease is one of the peripheral caused by the defect in peripheral tolerance mechanism, where you can it's caused also by commensal microbiota in the gut because of the strong immune reactions to these microbiota, which is normally not present there. But when you have Tregs in the, so what you're actually looking at is the cross section of the gut or the colon normally. So you have all these nice villi structures that are having a lot of goblet cells here, but when you have an inflammation, there's epithelial hyperplasia and cellular dysplasia that's causing all these cells to grow, and this is an instance of inflammation. But when you have Tregs, they don't have it anymore, showing that Tregs are really important to prevent this inflammation in the gut. Okay? And these are different forms of Crohn's disease, different forms of inflammatory bowel disease, Crohn's and ulcerative colitis, which are restricted to small intestine and colon respectively. and these are again caused by dysbiosis in commensal bacteria, and which subsequently causes the T-cell inflammation to ensue. So coming to the oral microbiota or the oral cavity, again, it's now considered that periodontitis is caused by absence of balance between resting bacteria and immune system. In a normal healthy tooth, so you have symbiotic biofilm and normal host microbe homeostasis and intact bone, where the host microbe interactions are in harmony, like immune reaction doesn't get generated to the normal commensal microbiota. However, in gingivitis and periodontitis, there is a dysbiosis, or there is a imbalance between the good bugs and the bad bugs, which is causing modest inflammation in the case of gingivitis and severe inflammation in the context of periodontitis. Okay, so although the exact nature of these interactions is still being studied, we know that it is strongly associated with dysbiosis of commensal microbiota and unnecessary inflammation by T cells. Okay? And it is believed that these pathogens, although they were considered to be the perpetrators or triggering bacteria of periodontitis before, now we think it's more the balance between these key pathogens and the good bacteria that's actually causing this inflammation rather than just the presence of these bacteria. Okay, so again, it's whether these are in symbiosis or dysbiosis that's causing homeostasis or overt inflammation, periodontitis. So how the microbiome is contributing to health and disease is attracting the interest of a lot of cell biologists, microbiologists, and immunologists, and our lab is also one. If you have questions, ask me, and this is this is why we need to have immune balance to tolerate harmless antigens and commensal microbes, and at the same time be effective at deleting pathogen microbes. Okay. So that's what the immune balance is about. If you have questions, please ask me now or email me later on. Yeah.
Okay, for autoimmunity dysfunction, is the problem primarily with the MHC on the cells that are being attacked, or the recognition systems of the attacking cells? Or is it a combination of both?
It's
We
a
talked about
combination
lot
of both. Yeah, it's a combination of both. Okay, so mostly the antigen, the healthy immune system knows that this is an innocuous antigen. We should not respond to this. But it's only the phenotype of these attacking cells that's different, that now recognizes this innocuous antigens, which is why the inflammation. So that's mostly the case, but sometimes it's also the combination. Yeah. Any other questions?
Can you clarify, because you were talking about the anti-allergen and anti-helminth, what is that?
Worms?
Oh, okay.
Yeah,
Gotcha.
yeah. Parasites can be toxoplasma, a lot of other things, but helminth, by helminth I meant worms.
Okay.
Yeah, and Th2 cells are responsible for eliminating the worms. Yeah. Sure. Okay, thank you. I thought I can give you a break and then continue with the rest of it, but then I thought maybe you need more break. So I'll let you go now. If you have questions, ask me, or you can go.